So welcome to this second class on microscopic organization of the digestive system. In our first class, we looked at microscopic organization of the whole GIT. And in this second part, we are looking at microscopic organization of the extrinsic glands or organs of the digestive system. Maybe just to remind us that when you look at components of the digestive system, we look at it in two entities. We have the whole GIT, which is the alimentary canal, the path followed by food. And that's the one we looked at last week. When, other than the whole GIT, the second component of the digestive system are the extrinsic glands or organs. What is important to note about the extrinsic glands or organs is that they are not incorporated within the gut wall. However, they have secretions which open into the gut lumen. These secretions are exocrine secretions, which means that they are released into ductal systems, which channel them to the epithelial lining of the alimentary canal. And so they are extrinsic in the sense that they are not incorporated within the wall. The glands which are incorporated within the wall of the whole GIT constitute intrinsic glands. And we mentioned some of them in the previous class. Things like the gastric glands within the mucosa, those are intrinsic. We have some salivary glands within the tongue or palate, they're intrinsic. The Brunner's glands within the submucosa of the duodenum, those are intrinsic. The crypts of Lieberkühn, those are intrinsic glands. So in this class, we are not looking at the intrinsic glands. We are looking at the extrinsic glands. And when you talk about these extrinsic glands or organs, there are majorly four that we're focusing on: the salivary glands, pancreas, liver, and gallbladder. And I want to look at them in that particular order. Let's begin with the salivary glands. They are called so because they produce saliva into the oral cavity. There are two types of salivary glands, the major and the minor salivary glands. The major salivary glands are the ones which qualify to be called extrinsic glands, and these are the ones we want to look at. They are paired glands. We have the submandibular, sublingual, and parotid salivary glands. So these are the ones we're going to look at in this class. And we've already done the gross anatomy of the same, so this lecture will just focus on their histology. The minor salivary glands are the intrinsic salivary glands. They are found within the wall of the oral cavity. So some may be in the palate, some may be in the cheek, some may be in the tongue. And so they go with those names: palatine, buccal, and lingual glands. Now, before we talk about histology of salivary glands, I thought it is important to bring to our attention some concepts that we look at histologically when we're describing glands. And I'll highlight key concepts which are relevant for this particular class. So generally, when you talk about a gland, remember a gland is a tissue, a group of cells within an organ that can produce and perhaps release substances that perform certain functions in the body. So whatever qualifies to be called a gland should be able to produce or release some substances that perform some functions in the body. A gland can be exocrine or endocrine. If a gland is exocrine, it means it produces secretions which are fluid in nature. Such fluids will be released via ductal system. There's usually a system of ducts that will receive these secretions And these ducts channel these secretions onto epithelial surfaces. And think about it. It could be an external epithelial surface, like the skin surface, like what sweat glands do, or it could be an internal epithelial surface, like what gastric glands do. They release their secretions through the gastric pits onto the surface of the stomach. So that is exocrine. Endocrine gland, on the other hand, produce hormones. Now remember, hormones are biological chemical messengers which are released into the bloodstream. And the reason why hormones are released in the bloodstream is because this chemical messenger need to travel to the target cell, but we need to enhance mechanism of its transport. And that is why it's released in the bloodstream. So hormones will be spread almost all over the body, wherever blood goes. And they'll find their target tissues wherever they are, and they can act on them. So glands can either be endocrine or exocrine. That's the message I'm making here. Now, most glands which are in part of an organ have a general structure. The general structure they have, and this does not matter whether they are endocrine or exocrine, is that any gland will have a stromal component and a parenchymal component. I need to elaborate what those two mean. When you talk about stroma, we are referring to the connective tissue elements of an organ. So this is the part formed by collagen fibers or other type of connective tissue fibers. And the main aim of the stromal component is to provide structural support to the organ or to the gland. Such stromal tissue usually will contain, in addition to just providing support, they'll be the one containing blood vessels that enter into this particular gland or organ. It will also be containing nerves. Most of the time, these nerves will be sensory in nature. And of course, they may also contain lymphatic channels. So the stromal tissue is a very important component of any organ. Yet, we then have the parenchymal component, which refers to the functional component. And if you're talking about a gland, then this is the secretory component. For exocrine glands, this parenchymal component will consist of the secretory units and the ductal units of that particular organ or gland. For endocrine, because endocrine don't have ductal system, it will just consist of the secretory cells. So generally, this is how we describe glands. Now, there's some things that you may need to then check when you want to histologically evaluate a gland. One of the things that you may just want to check is how the parenchyma of that gland is organized. For example, look at how the cells of the parenchyma are arranged. Are they in a cluster morphology, which means the cells are just lumped together? Or are they in a chord morphology, which means that the cells are arranged to form some linear structures? Or are they in a follicular morphology, which means the cells are arranged to form some larger, rounded, or spherical, or ovoid structures? If any of those three is true, then most likely the organ we're looking at is an endocrine gland as opposed to an exocrine gland. And perhaps we begin there by me asking you questions. So there's an image in your screen. I want you to tell me, based on what I've told you, would you classify that organization as cluster, cord, or follicular, and why? Anyone? Yes, Steve.
I would classify it as follicular because the cells are larger and rounded. Yeah.
Okay, very well, Steve. So yes, this is follicular morphology. I agree with you. The part I want you to, however, edit is that the cells are not larger and rounded. The cells are actually these dots you're seeing. But those cells are arranged around some structures which make it have those ovoid rounded appearance. The black dots are actually the cells, but they seem to be surrounding something. So this large structure is not a cell, but it's a structure surrounded by multiple cells. The whole entity is what you're calling a follicle. So yes, this is follicular morphology. Another image, the second image, anyone, would you classify this as a cluster, cord, or follicular? Yes, Winnie.
I'll classify it as cluster since the cells are closely packed and grouped together, forming like one continuous bundle. Yeah.
Very well. So yes, this is a cluster morphology. The cells are lumped together. It's difficult to describe it in any other word. Great. And therefore, the last one is definitely a cord morphology. This is cord morphology because we can see the cells form linear structures. Like that is a cord, that's another cord. So they're forming linear structures. So yes, if the morphology of the parenchyma fall in any of those three, then most likely what you're looking at is an endocrine gland. Now, what's the situation if it is exocrine? For exocrine morphology, the parenchyma tend to be arranged in a different way. They could either be of tubular arrangements, or it could be an alveolar arrangement, or it could be a sinus arrangement. And this is with reference to the secretory portion of this exocrine gland. We are not focusing much on the ductal portion. We are focusing much on the secretory portion of this exocrine gland. Now, let me show you some images of what those mean. So this is what you describe as tubular arrangement. If this is the arrangement, histologically, when this is cut in cross-section, this is how it will appear. So when you see something like this, we know that these are tubular arrangements of exocrine glands. You may also have something that looks like this. So this is the ductal portion, and this is the secretory portion. What do you notice? The cells of the secretory portion look like prisms. They have a wide base, and they're very tall with thin apex. And so the secretory portion of this structure is ballooned out. Although it's ballooned out, the lumen is very narrow because the cells are tall and tapering at the apices. If you take a cross-section of this is what you'll see. Important to note is that the lumen is very narrow, although it's ballooned out. So this qualifies to be the acinar morphology. And then the last one. Yes, the secretory portion is ballooned out, but the cells are predominantly simple cuboidal. And so the lumen of the secretory portion is also wide. If this be the scenario, then we call that the alveolar morphology. So I want you to have a graphic view of what these three look like in these drawings, because I want to now show you slides, histological slides with those varieties, and we should be able to pick them out. So this is tubular, this is acinar, and this is alveolar morphology. If a gland, the parenchyma of a gland, has any of those three arrangements, then that gland will qualify to be called an exocrine gland. Great. So let's look at these four. They all display the same thing. Question is, will we call this tubular, alveolar, or acinar morphology? Anyone? Yes, Anto?
Tubular morphology.
Yes, yes, you're right. This is tubular morphology. We can see that's a tubule actually with the lumen there. This is also tubular, and remember, we may have already looked at this one, this second image. So these are the intestinal crypts of Leverkusen found in the large bowel. They are tubular morphology. These are also tubular glands. We can see them cut in cross-section. And the last one are also tubular glands. These are characteristic of the sweat glands, which are tubular glands as well. Okay, the second one, anyone wants to try? Yes, Tamara?
These are alveolar glands.
Very well, these are alveolar morphology. We can see that the wall is simple cuboidal and the lumen are wider. And so that cuts across all the three. Great. And therefore, it goes without saying that the last one are the acinar morphology. Take note that in the acinar morphology, sometimes it's even difficult to see the lumen of the acinars. Like this one, the lumen is almost completely obliterated. A few of them, you may be able to see a very thin lumen, like for that one, you can see a small lumen there. Or in this case, most of them are obliterated, but that one has a small lumen, that one has a small lumen, that one has relatively larger lumen, or that one. But they're all acinar morphology. Perhaps these ones tend to have some bigger lumen, but they're all acinar morphology. There's a reason why we are calling this exocrine, the first one, other than the acini, and this is how acini look like, and I want you to have that graphic memory. We can also see a duct here. So this is a duct and not a secretory portion. The presence of a duct also tells you that you are most likely looking at an exocrine gland as opposed to an endocrine gland, and especially if that is mutually exclusive, to mean that the gland you're looking at does not have both functions. All right, so that is organization of the parenchyma. The other thing we want to look at, and especially if you have acinar morphology, and this becomes handy for this particular class that you're looking at. The salivary glands usually have acini. The pancreas also has acini. Now, when the secretory portion is of acinar morphology, then we need to go a step ahead and look at the types of secretory acini present. There are three options. The acini could be predominantly mucus. And remember or take note that I'm using the term predominant because most of the time you may not have one exclusive one. So look at the predominant morphology. So it could be predominantly mucus, could be predominantly serous, or could be seromucus morphology. If it is predominantly mucus, it means that the acinous cells produce a mucus secretion. How do mucus secretions look like on light microscopy? They tend not to stain. They don't pick color because the stains we use are most likely hydrophilic dyes. So they don't take them. So they'll appear clear. You see the cell outline, yes, but it's clear. The serous ones tend to stain with the hydrophilic dyes, and so they'll actually have a deep stain of the cytoplasm. Seromucus would mean that you have an almost equal, not exactly equal, but an almost equal proportion. Or let me use the term, you have significant contribution of both of them. So that becomes seromucus. Okay, let's look at this. So we have 4 images that I want to show you. This, that one, this, that one, this, that, and this, that last one. So I want to open it up for anybody who wants to comment on any of them. How would you classify which one? Yes, I'll start with Arman.
All right, so I can talk about the farthest to the left.
Okay.
And I would like to think that that is mucus because it's not picking up the stain well.
Okay, enough. Anyone else for same or another one? Okay, we'll go to Anrita. Anrita, you there? Okay, Anrita, can't hear you. So go to Ariana.
Okay, can you hear me?
Yes, raise your voice, Lakini.
Okay, I'll just correct the left one, the complete left one. I think it's a seromucus because you have both serous cells and mucus cells. So some of them are deeply stained while others aren't stained.
Okay, so I believe that would be correct. We go with that. This region here, this is a mucus. That is true, but this region here is serous. So this would qualify as seromucus. Yes, Winnie.
I'll go for the, I think also the second one from the left. I'll also classify it as seromucus since some parts are serous, they deeply take the stain, while other parts don't take the stain. So that will be classified as mucus, so seromucus.
Yes, you're right. So these are the mucus acini, and the rest are the serous acini. So that seromucus, I agree. All right, Michelle.
I wanted to do the third one, the one that's like a light purple. I think that's serous because you can see that it's picked up the stain.
Yes, so most of the acini here have picked up stain. So this is predominantly serous, and this is the ductal system of this particular gland. So this is predominantly serous. Yes, Papia.
So the last one, I feel like it is mucus because they're not picking up the stain.
Very well. So this is predominantly mucus because we can see the acini are not taking up stain almost at all. Now, if you look at it keenly, you'll notice that there's some things that may be taking stain. This is taking stain. So that is the ductal portion of this gland. Remember, what we are describing are the secretory portions. But the things that are taking stain here are the ductal portion. The other things that could be taking stain are these regions between the acini. Those are basically the stromal tissue elements of this particular structure. So the stromal tissue will still take up stain because remember, there are fibroblasts with some thin septa of collagen. Those ones may still take stain. But the parenchyma, the acina parenchyma that is predominantly mucus will not take the hydrophilic stains. Yes, Ariana, you have a question?
Yeah, I wanted to ask, on a histology slide, does the ductal portion always appear stained?
Most of them will. Most of them will, except if the ductal portion also has mucus cells.
Okay, thank you.
Welcome. Right. So we have mentioned a number of things regarding the things we need to check when you're looking at glands. And that is adequate to prepare the stage for what you want to learn today for salivary glands. So if you're looking at a salivary gland, what are the things that you will pick? First, the major salivary glands are encapsulated from what we learned from gross anatomy. This capsule is therefore part of the stromal tissue. And for most of them, again, this capsule sends in some septa into the gland that divide the gland into lobules. So grossly, you will see that the gland is lobulated as well. These are features that you would rather pick at the lowest magnification possible on your microscope. The connective tissue elements, how it's covering the whole organ and how it's sending in septa to divide it into lobules. Then we focus on the parenchyma. The parenchyma of salivary glands is of acini morphology. Within the acini, we usually tend to have some scattered adipocytes. Take note of that. So they're usually just scattered. And I mentioned because you may see them in some slides and you wonder, why are we looking at adipocytes? Usually, all of them may have scattered adipocytes. But we are focusing not on the adipocytes, we're just focusing on the acini. So they have acini morphology. Because they are exocrine glands, they have ducts. The ducts that they have could be within the lobules, so we call them intralobular ducts, or they can be between the lobules, which means they are outside the lobule. And then we call them extralobular ducts. The extralobular ducts are definitely much larger ducts compared to the intralobular ducts. The ducts will begin from inside the lobules, then they join outside the lobules. So extralobular ducts will be a union with some ducts which are formed from multiple intralobular ducts. And so they should be larger, much bigger. The larger the ducts, the thicker its wall. So extralobular ducts will then tend to have even stratified columnar epithelium on its wall. or they could be very tall columnar, simple columnar epithelium on the wall. The intralobular ducts, on the other hand, will tend to be more of simple columnar, sometimes simple cuboidal as well, because they're smaller. Take note that the intralobular ducts have narrower lumen and they're within the lobules, while extralobular ducts are much larger and they're outside the lobules. For us to distinguish between parotid, submandibular gland, and sublingual gland, we will focus largely on the type of acini. Parotid gland will be predominantly serous. Sublingual gland is predominantly mucus. And submandibular gland is seromucus. It's a mixed type. That becomes a major distinction of the three. Of course, if we had the advantage of the gross view, we then know that parotid is much larger compared to submandibular, and sublingual is the smallest of the three. But you don't have the advantage of viewing it grossly on a microscope. And so we'll rely largely on what I've just told you. You look at the type of secretory acini. Having said so, this is typical of how salivary gland will look like. What do we notice here? We notice some lobulations. So that's a lobule, that's another lobule, that's another lobule. With this region being the regions where we expect the stromal tissue passing and dividing the organ into lobules. Well, there are some blood vessels which are also seen. They are large vessels, but they are seen within the stromal tissue, as I told you, that the stromal tissue convey blood vessels. At a higher magnification, we note that there are ducts present. Look, these are ducts, for example. Because these ducts are inside the lobule, so these are intralobular ducts. We can also see adipocytes. Remember, adipocytes are usually very large cells with eccentric nucleus. They may exist single or in small groups, as you can see in these ones, but usually they're much larger cells. We can see serous acini, several of them. They are low magnifications though, but these are serous acini. Then we can see the mucus acini as well. This is still a duct. The red ones are most likely blood vessels. So that those that are associated with the ducts, still blood vessels. Okay, so typically this is what salivary glands will look like, a major salivary gland. I've told you that the distinction between parotid, submandibular, and sublingual will rely largely on the amount of serous acini and mucus acini that we see. Let's say something about each of them that we'd already mentioned in the gross lectures. So for parotid gland, the largest gland, and remember it lies lateral to the face at anterior to the temporomandibular joint and anterior to the ear. This is the parotid gland. It's the largest gland. The duct of the parotid gland traverse multiple structures and open opposite the second maxillary molar teeth. Remember, in terms of innervation, parotid gland receives its secretomotor innervation from the glossopharyngeal nerve, which is the 9th cranial nerve. The postganglionic fibers arise from the otic ganglion. The secretion that comes from parotid is predominantly serous secretion. How about the sublingual sublingual. Sublingual is located below the tongue, and it drains via multiple ducts into the floor of the mouth. Its secretomotor innervation is from facial nerve, and the relay ganglion is the submandibular ganglion, which is found just next to the mandible. This gland is predominantly mucus in its secretion. Now, take note of something. The farther a gland is from the oral cavity, the more its secretion will be serous, because serous secretions will not have struggle traversing that long path to reach the oral cavity. But you cannot have a predominantly mucus gland far away from the oral cavity. The risk of mucus impaction will be very high. So, sublingual is below the tongue. It's literally on the oral cavity proper. And so this will be a mucus gland. The mucus doesn't have to travel long distance to reach the oral cavity, while parotid, far away, can afford to be predominantly serous. And that gives way then for submandibular. That submandibular will be expected then to be a mixed gland. Located below the mandible, the duct of submandibular gland is called the Wharton's duct, which drains in the floor of the mouth via the sublingual sphincter. Remember, the innervation of submandibular is similar to the innervation of sublingual gland, and that this is the duct point of opening into the floor of the mouth. That's where it opens. Or in this image, this is the duct of the submandibular gland open. Submandibular gland receives, or rather, is a mixed salivary gland, which means it has both serous and mucus secretions, almost in significant proportion. I'll avoid the term 50-50 because it's not true, but significant proportion is a better terminology to use. Great. Having said so, I've already taken you through this, what we expect, that the gland will be lobulated. So the sections I'll give you may not have the advantage of showing you the capsule because are captured inside, but you'll be able to see the lobulations. And so I want us to look at the lobulations. If I show you a section that has a capsule, well and good. Remember, that's general for most glands. So it having a capsule doesn't categorically make it exclusively a salivary gland. It having a capsule just tells us it's most likely a gland or an organ. Fine. We need to find other reasons of what we think whatever you're looking at could be. So I've already told you this, that salivary glands will be lobulated, and that when you look at the parenchyma, we expect to see acini arrangement of the parenchyma, which can either be serous, mucus, or seromucus. So with that in mind, I want us to now try identify the three major salivary glands. And I'll start with this slide. So I have perhaps 3 or 4 levels of magnification. This is the lowest level of magnification, and I can help you here. So we see an organ that is lobulated, and I hope you now understand what that means. That's a lobule, that's a lobule, that's a lobule. We can see multiple lobules. We also note that the organ is encapsulated, and that's the capsule that we can see. So it's encapsulated. We can therefore categorically say that the capsule sends in septae or trabeculae that divide the organ into lobules. And we can also say with confidence that some of those septa carry what we can either consider as blood vessels or even ducts. Maybe you need a higher magnification to confirm that. So that is a duct magnification. Second magnification, we are now zooming onto one of the lobules. And yes, we confirm this is stromal tissue. So this could be one lobule if you look at it as one entity. I know we can still see some smaller parts. But yes, we can see the parenchyma of this particular gland at this level. There's another magnification that I want to give you. So I want to look at how this looks like and have a pictorial memory in your head. Then I give you the third magnification. So I've zoomed into one of the lobules much higher. And so we can see the acini. So question, what could this be? Anyone? Yes, Santo.
I'm thinking parotid
Okay, why would you think this is parotid?
because I'm seeing that it has acini that are serous.
Okay, I agree with you that the parenchyma is predominantly serous acini. I agree with you on that one. Yes, Bran.
Okay, in my opinion, as much as I think that as much as we have established this gland has some exocrine function, within the particular lobule or lobes, you can see some cluster of cells, some cells that appear to be clustered together. So I am not quite sure whether these will still be salivary glands, especially because we can see cluster of cells that are different from the acinar cells that have those basal nuclei. So I will actually say that this is a gland that has both exocrine and endocrine functions. And looking at the cluster of cells, this probably might be the pancreas.
Okay, very well. And you are right, this is not a salivary gland. And that was a message actually, that it's very easy to confuse parotid salivary gland with the pancreas, very easy. Because both of them will have serous acini. But there are some things that help us distinguish the two. And top on the list is the fact that pancreas will have, in addition to acini arrangement of cells, and let me show you the next magnification, in addition to the acini arrangement of cells, which we see in this particular zone, we also have regions where we have cluster arrangements. And this is a cluster. Those clusters are termed the islets of Langerhans. And the moment you see the islets of Langerhans, or at least the moment you see some clusters of cells scattered around or within the acini, then you know that we are not just looking at an exocrine, a pure exocrine gland. That's a major difference between pancreas and parotid. So yes, you're right, this is pancreas and not parotid, based on that. Okay, next. So this one is a salivary gland with confidence, and I'll tell you why. So at low magnification, we see a capsule. We also see that the connective tissue elements are still going into the organ of the gland, and that where we have these connective tissue elements, we can also see ducts. So these are the extralobular ducts. The extralobular ducts are between lobules So you can also call them interlobular ducts. Now, don't confuse inter with intra. So these are interlobular or extralobular ducts. And then this is now the parenchyma. A higher magnification of the parenchyma displays some scattered adipocytes, as we mentioned. But we also note that the parenchyma is predominantly serous. So if you remove the adipocytes, everything else that remains in front of you is actually predominantly serous. Don't confuse the adipocytes with the mucus acini. Take note that within the parenchyma, you also see ducts like that, and that one, and that one. So these ducts are within the lobules, so they qualify to be called intralobular ducts. Higher magnification displays the acini. So this is an acinus. That's another acinus. We confirm that it's very difficult to see the lumen of the acinus because of the shape of the secretory cells. We also confirm that this is a duct. So this being a salivary gland, predominantly serous acini, with both intralobular and extralobular ductal system, this will qualify to be called the parotid salivary gland. And you can confirm the findings I've already mentioned to you. We have intralobular ducts and extralobular ducts. We have predominantly serous acini with scattered adipocytes. So this is the parotid salivary gland. Maybe let me mention something about the ductal system. So we have extralobular ducts or interlobular ducts, which are very large ducts with stratified epithelium on its wall. Could be stratified cuboidal, stratified columnar, at least not stratified squamous. But you also have the intralobular ducts, which are smaller, like these ones. Take note that the extralobular ducts are within connective tissue stroma and perhaps surrounded by some blood vessels, as you can see here. The intralobular ducts, on the other hand, are within the lobules. Two types of intralobular ducts we have. There's an interlobular duct that is just forming from the acinus, like that one. The interlobular ducts that are just forming from the acinus constitute what we call the intercalated ducts. So the ducts which are incorporated within the acini, intercalated ducts. And then the intralobular ducts, which are fully formed, well-defined, are called striated ducts. Now, why striated ducts? Let's look at the basal zones of these cells. You may be able to see some linear striations, especially in the basal side, in that direction, from there, that direction, some linear striations, especially around this zone. Let me see if I have a high magnification. Yes. So you can see some linear striations there, especially in the basal zones of the cells. This demonstrates how the mitochondria are arranged in these cells, several arrays of mitochondria in columns. So because this is light microscope, you'll not be able to see the mitochondria, but we'll have a perception of what they give us based on their arrangement. They'll appear as striations, and so we call them striated ducts. The striated ducts usually have a lot of function in terms of active ion transport, and that is why they'll have a lot of mitochondria. Remember, striated ducts are part of the intralobular ductal system. All right. So that was parotid. I want to show you another gland. So this is also a salivary gland. Now, I've not shown you the capsule. I have gone direct into the inside. We can see a duct there. Based on the surrounding connective tissue here, I would call that an extralobular duct. But these other ones are intralobular ducts. At this magnification, I confirm the ducts that are there. I can see serous acini, but I can also see mucus acini. So these are mucus acini. These are serous acini. Take note that they're very different from their adipocytes. Higher magnification displays something very important about the submandibular gland. You can see a mucus acinus there. Then you can see a crescent-shaped serous cell around the mucus acinus. So this one here is a crescent-shaped serous cell around the mucus acinus. This is also crescenteric serous cell around the mucus acinus. If you think about it, there are actually very many, like that, like those ones. Even here, that's on these ones. We call these ones serous demilions. It means half a moon. They're not exactly half, but they're crescenteric. So submandibular gland is unique in two ways. One, it is a seromucous salivary gland, which means that's both serous and mucus acini. And then it has serous demilions. A serous demilion is an arrangement where you have a mucus acinus with a serous cell around its region. So the serous cell forms a cup around the mucus acinus. So that's what we call the serous demilions, very unique to the submandibular gland. The last one is definitely sublingual gland. Why do we say so? Yes, we can see that we see the connective tissue stroma. We can see lobulations. A higher magnification of it shows us that the acini are predominantly mucus. Maybe a much higher magnification will display that better. The acini are predominantly mucus. What you see staining here are the stromal elements. And so this becomes the sublingual salivary gland. So just use the concept of what type of acini are present to distinguish the three salivary glands. Great. I want us to also talk about the pancreas before I give you a short break. Now, pancreas is located behind the stomach, around the curvature of the duodenum, as we already know from gross anatomy. Grossly, it has multiple parts: the tail, the body, the neck, the head, the uncinate process. In terms of orientation, the tail is usually at a higher horizontal level compared to the body and the head. So when we do cross-sectional imaging, like CT scan, MRI, and we're coming from up, we'll start sectioning the tails before we section the body and then the head. And finally, the uncinate is the last thing we section. The pancreas has both exocrine as well as endocrine functions. The exocrine functions are basically in terms of production of pancreatic juice, which contain digestive enzymes and bicarbonates. The endocrine functions is with regard to the fact that we have islets of Langerhans that produce several hormones. There are several cell types within the pancreas. The cells of the exocrine pancreas would be the acinar cells, the central acinar cells, and I want to add another one, the ductal cells. Then the cells of the islets of Langerhans would be the alpha cells, beta cells, delta cells, and the PP cells. The other minor ones, I'll focus on those 4 major ones for the islets of Langerhans. So take note that the pancreas has 2 functional components, the exocrine and the endocrine component. Let's begin with the exocrine pancreas. So when you say exocrine, it means it's producing its secretions into a ductal system. The part that produces secretions into the ductal system consists of several acini. So they are acini in nature. The pancreatic acini are all over the pancreas, and they're the ones that produce pancreatic juice into the duodenum via the main and the accessory pancreatic ducts. In total, pancreatic acini constitute about 70% of the total pancreatic mass, so they constitute the largest proportion. The principal cells I've already mentioned are the acinar cells. So the acinar cells will be here, surrounding the acini. Those are the secretory cells. They largely produce the digestive enzymes. Then you have the central acinar cell. The central acinar cell represents the initial portion of the ductal system that enters into the acinas. So the ductal system of the pancreas begin at the center of the acinas. And so they constitute the central acinar cells. So that the acini of the pancreas will have the acinar and central acinar cells. The presence of central acinar cells also a distinguishing thing between the pancreas and the parotid salivary gland. Then we have the ductal cells. So the ductal cells will be the cells that line the ducts. The rest of the ducts other than the central acinar. These ductal cells are the ones which are largely helpful in secretion of bicarbonate-rich fluid. So the water component that has a lot of bicarbonate will largely come from the ductal cells. And then the secretion that are rich in digestive enzymes will largely come from the acinar cells. So I'd already shown you this slide of the pancreas. We can confirm that the pancreas histologically is encapsulated and it is also lobulated. At this magnification, that's what we're able to capture. Perhaps we can also see several tubular structures. At this magnification, we're not able to tell whether they're blood vessels or their ductal system. Maybe the next magnification will help us. Then we note that the parenchyma consists of acini, but we also have clusters of cells. These cell clusters could be light staining like what we see here, but they could also be deeply staining depending on how the slide you're looking at was prepared. Sometimes you may have trouble that they stain almost the same and it's very difficult to pick them, except your knowledge of a cluster and an acinus. So as you go to the lab, I'll want you to put a keen eye on what we mean by cluster arrangement and what we mean by acini arrangement, because that becomes helpful if the color differentiation between the islets of Langerhans is not very good from the pancreatic acini. So anyway, these are the acini. At higher magnification, we can see this is an islet of Langerhans, but these are the pancreatic acini. Now look, they are predominantly serous. We can see the lumen of acinus from what I told you earlier, that acini tend to have obliterated lumen. Now scan through and you'll see some cells at the center of the acini, like this one. So that is what qualifies to be called a central acinus cell. So this is one acinus, and that's at the center of the acinus. That's a central acinus cell. This is also a central acinus cell. This is one acinus, and that is a central acinus cell. So pancreas has the central acinus cells. Okay, these are still acini with the central acinus cells, as you can see in that region. So this is one acinus with the central acinus cell there. This is also a central acinus cell with the acini there. Great, so that is pancreas, especially the exocrine component. Now let's look at the endocrine component. The endocrine pancreas consists of clusters of hormone-producing cells that we call the pancreatic islets of Langerhans, and that's how an islet look like. Typically, it's a cluster of cells. So again, don't confuse them with some empty spaces within the pancreas. Like that's not an islet, that's an empty space, or most likely a blood vessel. Take note that the islets are heavily vascularized. You can see several blood vessels adjacent to them. The islets are usually found all over the pancreas, but most of them are concentrated in the pancreatic tail. That's why sometimes in surgeries like splenectomy, if the tail of the pancreas also goes, it may induce somebody to have diabetes. They constitute about 2% of the total pancreatic mass. I'm sure you're wondering where the 28 went. So the 28 is attributed to the stromal tissue of the pancreas. Remember, I mentioned that 70 is for the acini. There are various cell types of the islets of Langerhans. We can just talk about them shortly after I've shown you. So these are the islets. Again, they are isolated clusters of cells within the pancreatic acini, and that's how you capture them. That's how you describe them. You can be lucky if they're lightly staining. So there are several cell types here. Let's talk about the cell types. So one of the cell types are the alpha cells. The alpha cells, which are also known as the A cells, are the ones that produce glucagon hormone, whose major function is to elevate the blood sugar levels. The second cell type are the ones we call the beta cells, which are also known as the B cells. Now, don't confuse this B cell with the B cell of white blood cells. The beta cells produce insulin, whose role is to lower the blood sugar levels. Now, the mechanisms on how this happens, we learn more in physiology. The third cell type, the delta cells, which are also known as the D cell. Take note that we don't have C cells. The D cells produce somatostatin that tend to inhibit pancreatic secretions, predominantly inhibiting the exocrine secretion, but may also inhibit the endocrine secretions. And lastly, we have the PP cells, named from the hormone it produces, pancreatic polypeptide. These ones are also called the F cells. So again, take note, we don't have the E cell, but we have the F cell. Well, the E cells are present, except I'm not mentioning them here, but these are F cells. Now, pancreatic polypeptide may have a number of functions which may not be very clear. One of them could just be to slow down absorption of food. Could also be to stimulate some pancreatic secretions. Right, so that is pancreas. So I'll give you a short break before we start on the liver. Right, we can proceed. So we now go to the third organ, which is the liver. From gross anatomy, you have already covered that the liver is located within the right hypochondrium as well as the epigastrium. Its location, the right hypochondrium, is predominantly by the right lobe of the liver, and on the epigastrium, predominantly by the left lobe of the liver. Other than the right and the left lobe, we are aware that we also have the caudate and the quadrate lobes of the liver. There are other ways of discussing the gross anatomy of the liver in terms of segmental anatomy. So that is more of gross perspective. I'll not cover that here. Histologically, the liver consists of a number of cells, and I'll be telling you the cell types in the next slide shortly. But what is unique about how these cells are arranged is that they tend to arrange in a cord structure. So you have this cord-like arrangement of cells. Tells you a lot about how the liver functions. Most of those cells are known as hepatocytes, and I'll talk about them shortly. In terms of blood supply, and this becomes important in how we are going to describe the story of the liver, so that's why I'm mentioning it. The liver receives two sources of blood supply. Oxygen comes in through the hepatic artery, a branch of the celiac trunk. And then nutrients come in through the portal vein. So both portal vein and hepatic artery enter the liver and vascularize the cell types of the liver. From these two, we have now veins which come out of the liver. They are known as hepatic veins. And the hepatic veins take blood now to the inferior vena cava. So from here, remember the portal vein will be carrying nutrients from the intestines. It's responsible for about 75 to 80% of blood supply to the liver, while the hepatic artery is responsible for about 20-25% of blood supply to the liver. Histologically, the liver may have different morphologies depending on the animal. There's some animals that have the stromal tissue of the liver being very thick and therefore very perceptible histologically. An example is the one I'm displaying here, which has been obtained from a pig. So the pig liver have a lot of stromal tissue within it compared to perhaps the human liver or other types of mammals that may not have a lot of this connective tissue. I'm using this to demonstrate something that is still present also on the ones that don't have a lot of stromal tissue. It doesn't mean that they don't have stromal tissue, just that we're looking at the quantity of the stromal tissue, especially the septa that pass through the different components of the parenchyma. So for animals that have a lot of that, like the pig, we see that And so we see that this stromal tissue divide the liver into some smaller lobules that we're going to talk about shortly. This stromal tissue will contain with it the branches of the portal vein and the branches of the hepatic artery. So they accompany the blood vessels. You'll not most likely have the stromal tissue around the hepatic veins because hepatic veins drain from the center of the lobules. Like if this is one lobule, that's a vein draining from the center of the lobule. We call such veins the central vein. So central veins drain from the center of the lobule, then they join to form the hepatic veins that take blood to the inferior vena cava. But when blood comes in, it will come in along the connective tissue elements. So these are either the hepatic artery and the portal vein. Hepatic artery and portal vein, most likely that's a portal vein because of its size. That's most likely a portal vein again because of its size and location. So these are either portal vein or hepatic artery, the ones associated with the connective tissue elements. And then this is a central vein. It's not associated with the connective tissue element. That was a central vein, that's a central vein, that's a central vein. Now, what do we pick from that arrangement? The lobules tend to be hexagonal, most of them, not all of them. They tend to be hexagonal lobules. And that hexagonal lobular arrangement is a very important thing to pick for the liver, because one of the things that you're going to use to know that you're looking at the slide of the liver, when you see the hexagonal lobules of the liver. So that's a central vein, that's a central vein, that's a central vein, that's a central vein, because this is a lobule. But this is not a central vein because we can see it is within the connective tissue system. These are not central veins. At a higher magnification, now this is almost human. We don't have a lot of connective tissue separating the hexagonal lobules, but we can still be able to perceive them. Like that is one hexagonal lobule or almost hexagonal. They don't have to be mathematically correct hexagon because remember we've taken a section randomly in the liver. And so it may give us different orientations, but that's also another lobule there. So the difference between this slide and the previous one is that we're not seeing much of the connective tissue that separates the lobules, the hexagonal lobules. Some important aspects that I want to highlight. At the center of the lobule, we have a vein. We call that the central vein. This is a vein that drains blood from the lobule and eventually join to form hepatic veins, which take blood to the inferior vena cava. But within the connective tissue framework, that may either be a lot or little, we have other three structures. One, we have a branch of the hepatic artery. So you may want to call it hepatic arteriole. And two, we have a branch of the portal vein. You may want to call it the portal venule. And three, you have a component of the biliary ductal system. They are intrahepatic ductal systems. So you can call the bile duct or bile ductule, depending on its size. Those three components are running together. They tend to run together within the connective tissues that separate the lobules. These three that run together are otherwise termed the portal triad. So when we talk about the portal triad, we're referring to a portal vein or its branch, hepatic artery or its branch, and bile duct or its branch. The duct here will be an intrahepatic. It is something within the liver, intrahepatic ductal system. So a component of the biliary tree, a component of nutrients coming in, and a component of oxygen coming in, they tend to exist together. Again, when you look at a region that has a portal triad, it is possible to see 4 or 5 structures instead of just 3. That does not make it change its name. The issue here is that you may have a vein, portal vein that has just branched. So you could see 2 portal veins within a region that also houses a single hepatic artery and a single bile duct. That does not make it a portal quadrant or whatever. It is still a portal triad. We are looking at the functionality of the components. So the functionality of the components are there are always 3. And so we talk about the portal triads, irrespective of how many they are. Like in this region, you could be able to perceive multiple of them. The largest one will tend to be the portal vein. Then a smaller vessel will tend to be the hepatic artery. And then the distinction between the ducts and the vessels, that the duct is lined by columnar epithelium. So the columnar epithelium ducts within the triads are now the bile components, the biliary tree components. So I've told you how to distinguish the 3. The vein is much wider than the artery, but they're all lined by simple squamous epithelium, they're blood vessels. Then the duct is simple columnar. Irrespective of how many they are, they still constitute the triad because we're looking at the 3 functional entities rather than the 3 anatomical structures. Let's talk about, okay, so this image shows you the central vein, and it also shows you the portal triad. So this is a portal triad, and that's the central vein. It also shows you how hepatocytes are arranged, the cells of the liver. They tend to arrange in cord-like orientation. And perhaps you may perceive at this point, or maybe not yet, but you're going to perceive that the cords tend to radiate towards or away from the central vein. So look at that direction, radiating from here. Look at this one, radiating from there. Look at those ones. They radiate from the central vein. So the cells which are arranged in cords, the cords are radiating away or towards the central vein. The gap between the cords are actually lined by endothelial cells, and so they're blood vessels. Those gaps that you're seeing are actually blood vessels. They are the capillaries of the liver. We call them the liver sinusoids. The liver has very large capillaries that are known as sinusoids. So these are the hepatic sinusoids or the liver sinusoids. Now, the hepatocytes of the liver, the cells of the liver, are surrounded by some other delicate framework of connective tissue, specifically collagen type 3, the reticular fibers. And this image shows us that it has special stain for reticular fibers. We see a fine branching of collagen fibers staining dark here, surrounding the cords of hepatocytes or the cords of liver cells. So what you see here is a demonstration of how elaborate the collagen type 3 is in the liver. This will still demonstrate to you region of central vein, and this is most likely a region of the hepatic, of the portal triad. So take note that the liver cells are supported intimately by collagen type 3 fibers, in addition to the presence of collagen type 1 fibers that could be prominent that surround the hexagonal lobules. The collagen type 3 surround individual cell within the liver. The collagen type 1, which we saw, especially in the pig liver, will be between the lobules. Let's mention something about the cell types of the liver. The most abundant are the hepatocytes. They perform most of the liver functions, which you're going to learn in physiology. Kupffer cells are the macrophages within the liver. So they are cells of the reticuloendothelial system, so to speak. We have the hepatic stellate cells, also known as the cells of Ito, which are known to metabolize vitamin A. These are the cells that store fat, so they're the fat-storing cells. We do have the endothelial cells that will be found within the sinusoids, so they line the capillaries of the liver. And we also have the cholangiocytes. Cholangiocytes are the columnar cells that line the ductal system that contain bile. Having said so, let's talk about histological lobules of the liver at a greater detail. So I've told you that you have some hexagonal lobules, that the cells of the liver are arranged in terms of hexagonal lobules. The center of the lobule is the central vein, but the corners of the lobule contain the triad. So therefore, blood comes like this. Blood from the vein or the artery will come from here and drain that way. Even this one drain that way. Even that one drain that way. Of course, this one will drain that way, drain that way, drain that way. Because blood drained towards the central vein from the hepatic vein. Sorry, it drains towards the central vein, which forms the hepatic vein. It's coming from either portal vein or hepatic artery. The territory of the liver, the histological territory of the liver that is drained by a single central vein is what we call the classical lobule of the liver. And the classical lobule of the liver is the hexagonal lobule we've been seeing all along. So we call that the classical lobule. It is hexagonal. The 6 corners of the hexagon contain the triad. The center of the hexagon contains the central vein. These central veins are the ones that eventually join to form the hepatic vein that take blood to the IVC. Yet we have another one, which we call the portal lobule. The portal lobule is a triangle whose center is a single portal triad. And corners are the central veins. So what can we say is a portal lobule? The portal lobule represents the territory that is supplied by a single portal triad. So that territory will traverse 3 classical lobules, or at least 3 classical lobules in this particular view. So that is the portal lobule, the territory supplied by a single portal triad. Remember, it is triangular in shape. Lastly, we have what we call the hepatic acinus. We consider hepatic acinus a diamond-shaped lobule with the two corners, two opposite corners being formed by the central vein and two opposite corners being formed by the portal triad. The hepatic acinus represents the functional lobule of the liver. What do I mean by that? The other two are largely anatomical. This third one is largely physiological. It may not be a very accurate anatomical entity, but a very important functional entity of the liver. Why do we say so? Let's look at how blood flows through the diamond. So from the portal triad, and we can consider portal vein, for example. From the portal vein, blood will flow that way. Of course, it will also flow that way and that way. Let's consider here. So blood flows that way. So this other portal vein, the one found here, will also take its blood that way and that way. This helps us to define some 3 territories within this diamond on either direction. On the direction of that classical lobule and on the direction of this other classical lobule, we have 3 histological zones of the hepatocytes. Zone 1 here represents cells which are subjected to a lot of toxins from the portal vein, because portal vein carry everything from the intestines, including toxins. These cells are also subject to a lot of oxygen from the hepatic artery. And yet again, they are subjected to a lot of nutrients carried by the portal vein. The cells in zone 1 are highly metabolic. Compare that to cells in zone 3 near the central vein. These cells receive the lowest amount of oxygen. They receive the lowest quantity of toxins, and they receive the lowest quantity of nutrients. These cells are less metabolic compared to cells in zone 1. So zone 1 cells are most metabolic cells. Zone 3 cells are the least metabolic cells. Of course, zone 2 is an intermediate. So those are the histological lobules of the liver. If you look at it from this view, so this is a portal triad, you can see multiple luminous structures there. And this is a central vein. So zone 1 will be somewhere there, and zone 3 will be somewhere there. Right, that is the liver and its lobules. So let's finish with the biliary tree. Components of the biliary tree are many, and you've already covered that in gross anatomy, so I'll not take you through that per se, except mention that we have intrahepatic components and extrahepatic components of the ductal system. We also have the gallbladder with its cystic duct. The wall of the ductal system will just be similar to what we know about ducts. So nothing much about that one. I'll not show you a slide of that. However, I'll want us to focus more on the gallbladder. Okay, before I show you that, let me mention this. So the gallbladder is a sac-like structure, very wide lumen. Its key functions being to store bile, so that explains why it has a wide lumen. Its key function also being to concentrate bile. And so we'll expect some structural adaptation that entitle it to be able to concentrate bile. In particular, we will see that the epithelium is thin for transport, but also has a lot of microvilli for absorption. In terms of its storage capacity, let me mention, it will be sac-like, but importantly, the epithelium of the sac is highly folded so that it can stretch or can accommodate stretch when it's filled. Then we have the third function, which is to release bile when it's needed in the intestines. The act of releasing bile will involve the contraction of the wall of the bladder. And for that reason, we expect the wall of the gallbladder to have several smooth muscles. It will have muscular wall. Now, the image presented to you here may easily confuse you, and that's why I brought it here. This is not gallbladder. Why do we say so? So we see a structure, yes, it has what we can say several folds of the mucosa, agreed. And we've said that we expect that in the gallbladder. We also have what we can call thick muscular wall, agreed, what we expect the gallbladder. What is not constant, however, is the size of the lumen of these structures compared to what we expect gallbladder to be. We'll expect the lumen of the gallbladder to be much wider because a sac-like structure. This one is more almost a tubular structure. So even though we see folds and we see muscle on the wall, because it's a tubular structure, this is not gallbladder. Actually, this is fallopian tube. You can easily confuse it with the gallbladder. Now we can look at the gallbladder. Based on the fact that the lumen is quite large, it's almost impossible to get the whole gallbladder in a single slide. And so this is a part of the wall where the lumen will be on this side, and this side is exterior. At this magnification, some things we can pick that confirm that this is gallbladder. One is the several folds of the mucosa. I've told you that that's important to allow stretch when the gallbladder is filled. So this epithelium will stretch out. So it's important that it's folded to allow for stretch. So it's an adaptation for storage. Deep to that, we can see a thick muscular zone, which allow contraction when we release bile to the GAT. The rest of what we see here is basically adventitia or serosa. They're just the connective tissue that surround the outside. Nothing much about that. Higher magnification, what do we notice? We notice that the epithelium is simple columnar epithelium, and it's lining the folds of the mucosa. The simple columnar epithelium, as you can see in that magnification. So that is important to note that the epithelium is simple columnar. At a very high magnification, we confirm one last thing. The epithelium has a brush border, which means that it has microvilli that promote absorption. So remember, this is adaptation for concentration of bile. So in closing, I want you to remember some 3 important things about gallbladder histology. 1, a highly folded mucosa. 2, a thick, smooth muscular wall. And 3, simple columnar epithelium with microvilli. If you remember those 3, and of course you know what you have in mind regarding what the size of the lumen should be, you cannot miss gallbladder. Right, so that is it. This just shows you again the simple columnar epithelium with the thick muscular wall that we talked about. Very thick muscular wall, and the exterior is adventitia, nothing much about it. All right, so that is it. That will be the end of the lecture. I'll invite you to ask any question that you might have.